Hi everyone, I'm Preeta Filena and I'm going to be teaching you all about carbohydrates. In the next 5 minutes, we're going to cover what carbohydrates are, how they are classified, and their structural features, and why they matter in biology. Let's begin. Carbohydrates are one of the four major classes of biological macromolecules, alongside proteins, lipids, and nucleic acids. The name is derived from hydrates of carbon, which historically scientists thought these molecules were simply carbon combined with water. We now know that they are far more complex, which means they range from simple sugars to elaborate complex polymers with major roles in metabolism, structure, and cell signaling, which means they help communicate cells. Now let's look at the biochemical definition and general formula of carbohydrates. Biochemically, carbohydrates are defined as polyhydroxyaldehydes or ketones, which means they have multiple hydroxyl groups plus either an aldehyde or a ketone functional group attached to them. The general formula is CnH2ON, which reflects the historical hydrate of carbon idea. Let's take glucose for example. The formula is C6H12O6. However, if we can notice, not all carbohydrates follow this rule strictly. The deoxyribose in DNA is C5H10O4, which means it's missing one oxygen, and so on and so forth. To classify carbohydrates, they are classified into four major types: monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Now, carbohydrates are mainly classified by the number of monosaccharide units. So, monosaccharides are single unit sugars like glucose, fructose, galactose, and ribose, for example. These cannot be hydrolyzed further. Then there comes disaccharides. Disaccharides consist of two monosaccharides linked together by a glycosidic bond. For example, sucrose, lactose, and maltose. Next, we have oligosaccharides, which contain 3 to 10 units, such as raffinose or stachyose. And lastly, we have polysaccharides, which are long chains of carbohydrates that, for example, could be starch, glycogen, cellulose, are classic examples. This classification reflects both structure and function of each type of carbohydrate. Now let's take a look at specific examples for each of the classifications of carbohydrates. Let's look at monosaccharides and disaccharides first. Glucose, a monosaccharide, is an aldohexose and the primary cellular fuel. Fructose, on the other hand, is a ketohexose and the sweetest natural sugar found. Galactose is an epimer of glucose at carbon 4 and is a part of lactose. Ribose forms the backbone of RNA and ATP. Now, if we look at disaccharides, glucose plus fructose with an alpha-1,2 bond gives us sucrose. Whereas if we look at glucose plus galactose with a beta-1,4 bond gives us a lactose. And maltose is two glucose units with an alpha-1,4 bond. Note that the different glycosidic linkages determine the properties of the disaccharide. Polysaccharides mainly serve two major roles: energy storage and structural support. Starch is the plant storage form. It is a mix of amylose, which is linear with alpha-1,4 bonds, and amylopectin, which is branched with alpha-1,6 linkages. Glycogen, on the other hand, is the animal equivalent, which is stored in the liver and the muscle. It is even more highly branched than amylopectin, allowing rapid glucose release. Cellulose uses beta-1,4 linkages, which creates rigid, insoluble fibers forming plant cell walls. Chitin, which is generally found as cell walls in arthropod exoskeletons and in fungal cell walls, is made of N-acetyl glucosamine, which serves as a similar structural role in fungi and arthropods, like I said. Now, the structural aspects of carbohydrates. There are three major key structural components here. First, monosaccharides cyclize. The open chain form reacts with itself to form a hemiacetal or hemiketal ring, giving us pyranose or furanose structures. Second, it undergoes isomerism. D and L refer to the configuration at the highest numbered chiral carbon. Epimers differ at just one chiral center. Glucose and galactose are C4 epimers. Anomers differ at the anomeric carbon, giving alpha or beta forms. Thirdly, glycosidic bonds form through condensation reactions linking monosaccharides. The bond type, alpha or beta, and which carbons determine the properties of the resulting molecule. Carbohydrates majorly serve three functions. First, energy source, where glucose, like I said earlier, is a primary fuel for cellular respiration, yielding about 38 ATP per molecule through glycolysis, the Krebs cycle, and oxidative phosphorylation. Second, it gives a structural role, where cellulose gives the plant cell walls their rigidity, chitin forms arthropod skeletons, and peptidoglycan maintains bacterial cell integrity. Third, cell signaling and recognition. Cell signaling, where glycoproteins and glycolipids on cell membranes mediate cell to cell recognition, immune responses, and receptor signaling. If we look at the ABO blood group system, for example, it is determined by the carbohydrate antigens on the red blood cells. Now, why are carbohydrates important for humans on a biological level? Let's find out. Carbohydrates are indispensable in humans. The brain relies almost exclusively on glucose, consuming about 120 grams per day. Glycogen is stored in the liver and the muscle, roughly 400 to 500 grams. This provides rapid energy during exercise or fasting. Dietary fiber supports the gut microbiome health and digestion through indigestible fibers. The ABO blood group system, like I mentioned, is determined by carbohydrate antigens on red blood cells. And carbohydrate also intermediates like ribose 5-phosphate are essential for nucleotide synthesis and protein glycosylation. Now, how do we summarize and take away our key points? Carbohydrates are polyhydroxyaldehydes or ketones with the general formula CnH2ON. They are classified by chain length, which is monosaccharides, disaccharides, and polysaccharides. Structurally, they exist in linear and cyclic forms, with important isomerism including the D and L forms, epimers, and anomers, which are all linked by glycosidic bonds. Their functions span energy production, structural support, and cell signaling. In humans, they serve as brain fuel, glycogen reserves, dietary fiber, and blood group determinants. These fundamentals will help support your understanding of carbohydrates and their role in metabolism, molecular biology, and clinical biochemistry. Thank you for watching.